Good evening, and today's topic for today's discussion will be on nucleic acids. So to get started, let's define what a nucleic acid is. What are nucleic acids? So nucleic acids are basically just polymers, meaning these are large biomolecules which are made up of repeating units. In this case, in the case of nucleic acids, those repeating units will be nucleotides. So to define nucleic acids, simply we could say these are large biomolecules or biopolymers that store, transmit, or express genetic information of a cell and have nucleotides as the monomer units. So there are two types of nucleic acids. These are DNA and rRNA, deoxyribonucleic acid and ribonucleic acid. So these two nucleic acids, DNA and RNA, differ in their structure, as we will see as we go on. But before we can talk about how DNA and RNA differ in structure, we can talk about the monomer units of the nucleic acids, the nucleotides. So nucleotides have three components. There are three components that will make up a nucleotide. You have a pentose sugar, and then you have a nitrogenous base, and you have a phosphoric acid or a phosphate group. When these components come together, they form what is known as a nucleotide, which is the structure shown here. So what is a pentose sugar? So pentose sugar is basically a monosaccharide, which is a carbohydrate that has 5 carbon atoms. So you have 1 there, 2 there, 3 there, 4 there, and the 5th carbon there. And this is what makes this a pentose sugar. So in the case of nucleic acids, the pentose sugar is either ribose or deoxyribose. The difference between the two is on the second carbon atom. So for the deoxyribose sugar, you have the first carbon there, and then on the second carbon, instead of having a hydroxyl group, you have a hydrogen. The oxygen will be missing there. Whereas in the ribose sugar, on the second carbon, you have that hydroxyl group. So if you compare the two structures, you have a hydroxyl group here, and there you don't have the hydroxyl group. You just have a hydrogen. The oxygen is absent in deoxyribose sugar. These two sugars are one of the differences between DNA and RNA, in that you have RNA having the ribose sugar, whereas you have DNA having the deoxyribose sugar. The absence of the oxygen on the second carbon makes DNA more stable than RNA. The nucleotides also have a nitrogenous base attached to them. These two structures here show the basic structures of nitrogenous bases. So you could have them having 2 rings, where they're known as purines. So you can remember that by simply adding purines to 2 rings. And then you have the one with 1 ring, which is known as a pyrimidine. So you have these combining with the pentose sugar and the phosphoric acid, and that is what will form the new nucleotide. So these nitrogenous bases can be one of 5 nitrogenous bases, So these nitrogenous bases are adenine, guanine, cytosine, thymine, and uracil. So adenine and guanine will be the purines, whereas cytosine, thymine, and uracil are pyrimidines. When it comes to naming nucleotides, they're named according to the nitrogenous base contained in the nucleotide. So if you look at the structures shown in this slide, in this structure, this nitrogenous base is guanine. And so this nucleotide will be guanosine monophosphate. Monophosphate in that it has one phosphate group here. If there were two, it would be guanosine diphosphate. If it was three, it would be guanosine triphosphate. So the full name of this structure is guanosine 5' monophosphate. 5' meaning the single phosphate group is attached to carbon number 5 of this ribose sugar. So you have 1, 2, 3, 4, then carbon number 5 there. And this is what this 5' over here is. So you have guanosine 5' monophosphate. The structure here, this nitrogenous base is adenosine, and then this pentose sugar here is deoxyribose. So this structure will be adenosine 5'. This will be deoxyadenosine 5' monophosphate. So this brings us to our next point. When you look at the nucleotide, as mentioned, the nucleotide will have three components: the phosphate group, the pentose sugar, and the nitrogenous base. When you remove this phosphate group, what you're left with is known as a nucleoside. So you have the pentose sugar, and then you have the nitrogenous base. And when these undergo a condensation reaction, so this condensation reaction is just the fusing together of the pentose sugar and the nitrogenous base with the loss of a water molecule. So you have the OH there and the hydrogen there on this nitrogen. You have the hydrogen there. So you lose this hydrogen and you lose that water molecule. And then this structure will fuse here, forming a nucleoside. This nucleoside will then go on and to condense with a phosphoric acid, and that will form a nucleotide. So when it comes to naming of nucleosides, they're also named according to the nitrogenous base contained in the nucleoside. In this case, we have the nitrogenous base being cytosine. So the nucleoside formed will be cytidine. In this example shown in this image, the pentose sugar was deoxyribose, and as such, the nucleoside was named deoxycytidine. If the nitrogenous base is uracil, that would be uridine. If the nitrogenous base is thymine, it would be thymidine. So the bonding I was talking about earlier on, where the pentose sugar and the nitrogenous base fuse, the bond that is formed is a beta covalent glycosidic bond. The glycosidic bond is simply a bond between a carbohydrate and another molecule. The other molecule could be a carbohydrate, or it could be something else, in this case, a nitrogenous base. So you have that there, the glycosidic bond, which is shown here. So this bond is known as a beta covalent glycosidic bond. And so the sugar molecule is bonded to the nitrogen atom, which is at the beta position. And this is what I was trying to explain in this slide by saying formed by the condensation, the nucleoside is formed by a condensation reaction, resulting in a beta covalent glycosidic bond. So this is the beta covalent glycosidic bond. This slide here just shows what I've been talking about, where you have a nucleoside reacting with the phosphoric acid to form a nucleotide. That's our phosphoric acid, and this is our nucleoside, guanosine. When the two react, you will form the nucleotide. So you have this condensation reaction here, where you have this OH here and this hydrogen here. They will leave as water, as a water molecule, and what you have is the fusion of the phosphoric acid with the nucleoside, forming a nucleotide. So you can have a situation where you have enzymes that will come and they'll add to this nucleotide. In particular, they'll add to this carbon here, the third carbon in the pentose chain. So it's the 3' carbon. So they'll add to this. And when they add another nucleotide to this carbon here, what you have is a dinucleotide. Another nucleotide can be added to the dinucleotide, forming a trinucleotide. So you have a set of enzymes that will come in and they'll continue adding nucleotides, bonding them together, so on until a certain number is reached that is required. And this polymer that is formed by the addition of nucleotides together is what a nucleic acid is, and it can either be DNA or RNA. So the required number for DNA and RNA will vary according to organism. For humans, for example, the human reference genome is about 3 billion or more base pairs. And so DNA in itself is a very large molecule. This slide here is just trying to show what I was talking about, the formation of dinucleotides and nucleotides. You have in this diagram the phosphate, the sugar, the pentose sugar, and the base coming together to form a nucleotide. And then in this diagram here, you have the nucleotide being combined with another nucleotide to form a dinucleotide, which is what is also being shown here. And as I mentioned, the connection between the two nucleotides will happen at the third carbon. The third carbon to be between the third carbon and the fifth carbon of the next nucleotide. And this linkage here is known as a phosphodiester bond or phosphodiester bridge. This brings us to the next section of our discussion, which is on DNA. And in order to understand DNA, we need to know a few things about its structure. So the DNA structure was derived by Watson and Crick. But before we talk about Watson and Crick, we'll talk about the Chaga rules or the complementary base pairing rules. So in the 1940s, scientist named Erwin Chagaf did chemical analysis on DNA, and his findings were that the total number of purines is equal to the total number of pyrimidines, and more specifically, the number of adenine base pairs was equal to the number of thymine base pairs, and the number of guanine base, the number of guanine nitrogenous bases was equal to the number of cytosine bases, and that these were bonded together by hydrogen bonding. So from the structures of adenine and thymine, you have the oxygen on the thymine interacting with a hydrogen on the amino group of adenine. And then you have this hydrogen bonded to the nitrogen on the thymine, interacting with the nitrogen in the adenine group, forming two hydrogen bond linkages. Whereas in the guanine and cytosine, you have for cytosine and guanine, there'll be a formation of three hydrogen bonds. So this hydrogen bonding between the base pairs is what is referred to as complementary base pairing. So the structures of the bases sort of complement each other in such a way that they allow for the two bases of the nucleotides to interact, forming hydrogen bonds. So for adenine and thymine, you have two hydrogen bonds being formed, whereas for guanine and cytosine, you have three hydrogen bonds being formed. So after his chemical analysis, Chagaf came up with these rules, and he also went on to say adenine cannot bond with guanine, and thymine cannot bond with cytosine. So another thing that was of importance to the determination of the structure of DNA was the Wilkins and Franklin experiments. So Wilkins and Rosalind Franklin carried out X-ray crystallography on DNA. They simply just passed X-rays through a DNA molecule, and what they found was that the DNA molecule had two strands. It was a helical structure, and that the nucleotides were stuck together with a spacing of 3.4 nanometers between them, and the complete 10 of the helical structure contained 10 nucleotides, and the whole DNA molecule, its diameter was 20 nanometers. I'll show this as we go on. So now comes Watson and Crick. So Watson and Crick, they used the results from the Wilkins and Franklin experiment, as well as Chagaf rules, and using those results and the rules, they made a model representative of the DNA structure using wires and some flat materials. So this is Watson-Crick in their model, which was made of wires and flat materials. So this model, representative of the DNA structure, has been represented here in a schematic diagram. So what Watson-Crick found was that you have the nucleotides in DNA, the nucleotides base paired to each other in this manner, forming this helical structure shown in this other picture. Then if you get that point there from the helical structure, so it comes which is somewhere there, comes again once it completes the 10. So the distance from that point to that point, they determined it has been 34 nanometers, which was representative of 10 nucleotides. Okay. So maybe this might be a better diagram. So say if we came from this point up to somewhere at that point, that would be 180. And as it comes up to this point here, this would now be 360 degrees, a complete 10. So if you take the distance from there to there, that would be 34 nanometers. And then the bases with, according to the model, were stacked together. So you have these base pairs here, and then you have the other base there, the base pairs there. Then you have another base pair there. It was so they are on top of each other, and so the distance between the base pairs, this distance here, was 3.4 nanometers, which interpreted in this 34 nanometers, the complete 10, having 10 nucleotides. So according to Watson-Crick's model, DNA is a double helix. It has two polynucleotide chains. It has 10 nucleotides per 10 of the double helix. And then the strands are antiparallel. What this simply means is that due to the complementary base pairing, you have one strand that will have, let me see if I can show this. Okay. So you have, if this was a DNA molecule, so you have this 5' end, the 5' carbon, and then you have the 3' carbon here, and then the nucleotide is connected. Then you'd have another chain this side. Instead of running 5' to 3', it would be running in the opposite direction. So you'd have the 5' nucleoside over there, and then the 3' nucleoside over there, and so on. So the Crick also, as mentioned, they also stated to say the bonding in the DNA molecule was by hydrogen bonding, but there were also other forces that were keeping it that were keeping this DNA molecule together, and these forces also include van der Waals forces and the hydrophobic effect. So due to the stacking, of the bases in the molecule, this distance between them, the 3.4 nanometers, is just enough to allow van der Waals interactions between the base pairs. And then due to, if I can use this other diagram here, so you have this strand coming here, and then you have the other strand coming like that. On the outside of the strand, you have the phosphate sugar backbone. So you have the phosphate and you have the sugar there. So due to these phosphates, which are charged molecules, what you have is that in the aqueous environment, which is the cell, you have water there. These phosphate groups will interact. They will interact with the water molecule, whereas the bases, which are formed hydrogen bonds with each other and are not polar, will be on the inside. So these charges on the nucleotides will create what are known as hydrophobic effects, where the bases are on the inside and not interacting with the aqueous environment. So that brings us now to the other nucleic acid, which is RNA. But before we proceed, as a quick recap of what we've mentioned so far about DNA, we've mentioned that its structure was determined by Watson and Crick. They used X-ray crystallography results from Wilkins and Franklin's experiments, as well as Chargaff's rules, and they worked out a model. And according to their model, DNA had a double helix structure, which was held together by hydrogen bonding of the base pairs. So according to Watson and Crick's model, the DNA molecule was a double helix. It had two polynucleotide chains held together by complementary base pairing. And apart from the hydrogen bonding, it was also held together by van der Waals forces due to the stacking of bases. And then also hydrophobic effects due to the polar nature of its backbone and the non-polar nature of the bases. So how does DNA differ from RNA? RNA will have a single strand, unlike DNA. In terms of sizes, RNA will be smaller. Why does it allows for RNA to be used to make proteins instead of DNA? And because of that, you have RNA being made from DNA, which is in the nucleus, by a process known as transcription. Therefore, RNA can be said to be an intermediate nucleic acid, in that it will act as an intermediate between the DNA and the expression of the DNA, which is basically formation, the synthesis of proteins. Of note also is the difference amongst the differences between RNA and DNA. Earlier on, we had mentioned of the pentose sugar, where RNA will have ribose and DNA will have deoxyribose. Another difference in the structure between the two would be in the bases. You have adenine, guanine, cytosine, and thymine in the DNA molecule. But the difference with the RNA molecule is that instead of thymine, the RNA molecule of uracil. Another difference between DNA and RNA is that whereas you have DNA having only one form, which is that double helical structure, for RNA, it will have variant structures, which gives rise to the three types of RNA. And these three types of RNA are messenger RNA, ribosomal RNA, and transfer RNA. And we'll look at how the different structures of these three types of RNA. So the types of RNA, you have messenger RNA, which can also be written as mRNA. So this one is just single-stranded. It's a single strand. So this will be made from a DNA molecule, a DNA strand. So you have a DNA molecule, a DNA strand. One of the two strands in the double helix, and some enzymes will come in, and then they'll, using the bases of this DNA molecule, they'll make a complementary RNA molecule. These enzymes are known as RNA polymerase. So you have those complementary base pairing forming an RNA molecule. So if this DNA molecule was say 5' to 3', the RNA molecule that would be formed here would be anti-parallel to this one. So it would run 3' to 5'. So this synthesis of the RNA molecule will occur in the nucleus of the cell. And once that messenger RNA has been synthesized, it will move into the cytoplasm, where it will go on to code for the protein synthesis. This is where RNA comes in, ribosomal RNA. It's also single-stranded, whereas mRNA will be synthesized in the nucleus. rRNA is synthesized in the nucleus. So you have this here, which is the nucleus, and then you have a part on the nucleus known as the nucleus, and this is where the rRNA is synthesized. So this rRNA will combine with proteins and to form a ribosome. Shown here is the structure of ribosomal RNA. when it contains two subunits. So you have a larger subunit and a smaller subunit. The larger subunit differing in sizes between prokaryotes and eukaryotes. So this whole unit, when it comes to the whole ribosomal RNA, when it comes to prokaryotes, will be 70S. So this 70S, what it simply means is that if you were to get you were to extract ribosomal RNA and centrifuge it, and using density gradient separation, you would have it at 70. You would have it at 70S. So this S here is what are known as self-bag units. Those are just units for after you centrifuge a particle and then it coordinates, which is just separates from other molecules in that substance, and then it settles at some level. So after it settles, the unit as well as the president unit. So in prokaryotes, it was found that after centrifugation, the ribosome would have a 70S. Then what they further did was that they centrifuged the large unit alone, and they found that it settled at 50S. And then when they centrifuged the smaller unit, it settled at 30S. So in prokaryotes, you have the 70S ribosomal RNA, which is made up of the 50S larger subunit and the 30S smaller unit. In eukaryotes, however, the whole ribosomal RNA was found to be 80S. And then the larger subunit was found to be 60S, and the smaller subunit was found to be 40S. So what is it about the ribosomal RNA? Apart from that, the one other thing worth knowing about the ribosomal RNA is that after it combines with certain proteins and forms what are known as ribosomes, and this is where protein synthesis occurs. And you have these ribosomes often occurring in clusters known as polyribosomes. So you have mRNA, ribosomal RNA. We are yet to talk about transfer RNA, tRNA. These three forms of RNA will interact together, as we will see in follow-up tutorials. These three forms of RNA will often interact with each other in the synthesis of proteins. So that brings us now to the third type of RNA, which is the tRNA, transfer RNA. So this tRNA is like all other RNA molecules, usually single-stranded, and it acts as an adapter molecule in that in the process of protein synthesis, as mentioned, it will interact with other RNA molecules to synthesize proteins. And what tRNA does is that it will transfer. There will be various tRNA molecules, each containing an amino acid, and they'll be interacting with both the ribosomal RNA and the messenger RNA. And through this interaction, they'll be synthesizing proteins. So the structure of the tRNA, this is the structure of tRNA. So it will have 4 arms. So we have the amino acid arm. This is the arm that will contain amino acids during the protein synthesis process. And then you have the T arm, and you have the D arm. And then at this far end here, you have the anticodon arm. And when you look at this structure, this structure is it looks similar to, there's a flower, if I can call it that. So if you can see, the structure of the tRNA forms a clover leaf shape. Yes, so this here, this is a clover leaf. That's what the structure of tRNA has been described as. And at this far end here, you have the anticodon arm. This anticodon arm will have bases there. These bases will be complementary to bases on a specific mRNA. So during the whole protein synthesis, we'll explore more when we talk of translation. So you have an mRNA which will have bases there as well, and these bases will interact with bases on the anticodon arm, and as a result of this, what you have is that tRNAs will only be able to bind to certain portions of an mRNA, and through interaction with the ribosome, there'll be that formation of the proteins, which are basically chains of amino acids. So the tRNA will have amino acid attached to an amino acid arm, and this amino acid arm will always end So this amino acid arm will always end in this sequence here. CC AA at the 3' end of the tRNA. So as a way of recap, we've talked about nucleic acids. We defined them as being polymers made up of nucleotides. And then we talked about how the nucleotides are formed. And then we talked about the two types of nucleic acids, DNA and rRNA. Them being nucleic acids, we said they carry genetic information. In our definition of nucleic acids, we mentioned to say nucleic nucleic acids are large biomolecules or biopolymers that store, transmit, and express genetic information. And this is true for both DNA and rRNA. They both contain genetic information. And then we've mentioned them being nucleic acids. They're made up of nucleotides. Both are present in prokaryotes and eukaryotes. And then talked about the polymerization, which will result in the formation of the DNA or RNA strand, where you have enzymes adding nucleotides one after another until a certain number is reached. And there's this point here that says cleaved by nucleases. So nucleases are just enzymes that are capable of breaking the bonds that hold the nucleic acid together. So they'll break that beta covalent glycosidic bond, and others will break hydrogen bonds in DNA. And then we looked at DNA on its own as a nucleic acid and RNA on its own as a nucleic acid. And this table here shows the differences that we've already discussed. So DNA stores and transmits genetic information, whereas RNA transmits genetic information. As we mentioned, you have RNA being made from DNA, and then you have proteins being made from RNA. So this process where proteins are made from RNA is what this statement means here when they say many transmits genetic information. So DNA stores and transmits genetic information. It transmits in that the RNA will have to be made from the DNA. Then it stores in that. So I've shown this diagram here. So you have DNA forming RNA by a process known as transcription, and then RNA forming proteins by a process known as translation. Then you have DNA, which is capable of making another DNA molecule by replication. So here, during this replication process, which we'll look at in tutorials that will follow up, that maintains the structure of DNA, and that is what they mean when they say DNA stores and transmits genetic information. And we looked at the structure of DNA, and we said the structure is double-stranded, whereas that of RNA is single-stranded. It may take different forms, but it still remains single-stranded. This statement, note it says mainly single-stranded, and here it says mainly double-stranded. So it's because for viruses, you can have there are certain viruses that will tend to have double-stranded RNA instead of single-stranded RNA, and you can have other viruses that will have single-stranded DNA instead of double-stranded DNA. And one of the other differences between DNA and RNA is that DNA is chemically stable, whereas RNA is chemically unstable. This is due to the fact that DNA, the double-stranded nature of the structure, and the hydrogen bonding, the van der Waals interactions, and the hydrophobic interactions, those will keep it. Those are what make it stable. Whereas RNA, which is single-stranded, won't have those interactions, and so it will be able to react with other substances that are capable of interacting with it. And we also went on to mention, to say, apart from the difference in the pentose sugar, DNA and RNA also differ in the bases found in the nucleosides. So for DNA, you have thymine, whereas for RNA, you have uracil. So you can't find thymine in RNA, you can't find uracil in DNA. And then also mentioned to say DNA is huge in comparison to rRNA. And this is why rRNA is used to is used in the synthesis of is used to code for the synthesis of proteins instead of DNA. And then we also mentioned to say DNA will be of one type, whereas for rRNA you have three types, which will differ in their structures. So we have ribosomal RNA, you have transfer RNA, and we have messenger RNA. So that brings us to the end of our discussion. I hope it has been helpful and hope to see you in the next session. Thank you.